Hello, my fellow viewers. Welcome to MR.Wanyoike, the learning digital platform. In front of you is MR.Wanyoike himself, ready to present you a biology topic in form 4, that is genetics. And we are on continuation of the previous where we left previously. And as far as genetics is concerned, we had covered several areas of genetics. And in today's lesson, I'm going to give you the reasons why the Gregory Mendel was very successful in his work. Therefore, reasons why which contributed to the success of Mendel. And the first reasons I'm going to discuss about reasons for success of Mendel. This is the father of inheritance, and the reason number one, he used favorable materials. He used favorable materials. He used favorable materials. And number two, as far as number two is concerned, the material he used by the name the pea plant. The pea plant he used, the pea plant he used has observable various. I can talk of has various observable and contrasting trait or characteristics, traits. And the number three, he focused on only on particular traits. And number four, he kept accurate data. He kept accurate data. These are the four reasons. Why he was very successful. And therefore, as far as Mendel work is concerned, the first reason why he was very successful to his work, as far as his success is concerned, the first one is because he used favorable materials. And favorable materials that he used a garden pea plant. And garden pea plant was very favorable because it normally which is self-fertilized, and therefore Mendel could employ cross fertilization at will. That is the first reason that contributed to his success. The reason I've said he used favorable material, and that is the garden pea plant, and because they are normally self-fertilized, and therefore he could use cross fertilization at will. And number two, the second reason why he was very successful is because the pea plant he used has various observable and contrasting characteristic. For example, you can give the characteristic of length. We have the short ones and the long ones, and therefore and also the color. We have some have a different color from the other. And as far as colors are concerned, I'm going to highlight all of them in the course of this topic. And as far as the third one is concerned, Mendel only focused on particular traits only, unlike the previous scientists who focused on the wholesome heredity of each organism. And therefore, those other scientists who had started the work of genetics before Mendel, they were focusing on the wholesome heredity of each organism, and therefore that's why they failed. Unlike Mendel only concentrated or focused on the particular traits only, as far as the inheritance is concerned, inheritance of traits is concerned. And lastly, the last thing that enables Medel to be very, very successful in his work is because Medel was able to keep accurate data, and therefore he kept accurate data, and therefore he used this data and analyzed this data to come up with a hypothesis, and these hypotheses are the ones that enabled him even to come up with the law of heredity. And these are the 4 reasons why Medel was very, very which contributed to the success of Medel, And as far as it is concerned, we are going to go to the next point. We are going to go to the next point, and the next point which I'm going to discuss with you today is what about the scientists today? We ask ourselves, what about the scientists today? What are they using? During the study of genetics? As far as the scientists of today's are concerned, they normally use a common fruit fly. There is an insect that we refer to as a fruit fly, and the fruit fly, the scientific name of fruit fly is called Drosophila. That is Melanogustus. Melanogustus. That is the scientific name. That is Drosophila melanogustus. Drosophila melanogustus. And as far as this fruit fly is concerned, it normally have various contrasting characteristics. And the first one, what are the various contrasting characteristics of Drosophila melanogustus? The first one, we have the wing length, and some of them have long wing, which is dominant over the vestigial wing, and therefore we can talk of the first one, the length of wings. We have the dominant wing is the length, that is the longer one, the long wings, and also we have vestigial wings, and the second one, we have the eye color, or the color of the eye, and therefore there are some of them have red eyes, and the others normally have white eyes. And the third characteristic is the size of the abdomen. Size of abdomen. Some have broad abdomen, while others have narrow abdomen. And the fourth one, we can talk of body color. That is the fourth one. And as far as body color is concerned, we have gray body color, and the others have black. We have black ones, black body color. And as I explain these characteristics, as far as the length of the wings is concerned, the ones that have long wings, the long wings trait is dominant over the vestigial wings, and therefore it means that in presence of the gene that determine the long cara, if they appear, if the allelic gene have one of them is dominant, which will be responsible for the bigger increase in length of their wings, it means that in presence of the dominant gene and the recessive gene, the individual or the fruit fly will have long wings, because as far as the long wings are concerned, that trait is dominant over the vestigial wings. And as far as the color of the eye is concerned, those fruit flies that have red color, the red eyes, it means that a red eye is controlled by a dominant gene, and therefore in presence of a relic gene where they are identical, such that there is a gene that represents the responsible for red color and another gene which is responsible for white color, the individual fruit fly will have red eyes, because the red eye, the gene that determines the red eye is dominant over the gene that determines the white eye. And as far as the fourth one is concerned, that is the size of the abdomen, abdomen. And as far as the allelic gene is identical, is not identical, I mean, whereby we have a gene that determine a broader abdomen, and there is also a gene that determine narrow abdomen. It means that that individual or fruit fly will have a broad abdomen. Why? Because the gene that determine a broad abdomen is dominant over the gene that determine the narrow abdomen, and therefore the gene that determine narrow abdomen is a recessive gene. Having said that, what about the body of the cara? There are two colors. There are two colors as far as the body color is concerned. That is gray, and the second one is black. And therefore, as far as gray, the gene that determines the gray color is concerned, is dominant over the gene that determines the black color. And therefore, in presence, if the allelic gene have an identical genes, that is the gene that determines the gray color and the gene that determines black color, it means that the individual fruit fly will be gray in color, because the gray color is the gene that determines the gray color is dominant over the gene that determines the black color. Having said that, I'm going to the next area, and the next area I'm going to discuss about the reasons why fruit fly is very important during the study of genetics. What are the reasons? What are the reasons why fruit fly, that is Drosophila melanogasters, why Drosophila, that is fruit fly is very important during the study of inheritance or genetics. We can talk of genetics. Why is it important during the study of genetics? That is a question you can ask yourself. That is a question. Question one. Reasons. What are the reasons? You can be asked in an exam. Give the reasons why the fruit fly is very important. Why it is very important during genetics or the study of genetics. The first reason, I'm going to highlight them, then I will discuss them. The first one is the female lays many eggs. The female lay many eggs. And number two, the second reason, have many. The fruit fly have many observable and contrasting. Observable and observable. We can talk of observable distinct or observable characteristics. In observable characteristics. And number 3, I'm going to explain them later. It is easily bled in the lab. It is easily bred. It is easily they are easily bred in the lab. And number 4, we have it has a short generation time. And number 5, offsprings can be backcrossed. Then backcrossed. And number 6, the flies are safe to handle. The flies are safe to handle. So these are the 6 reasons why it is very, very important. The reasons why the fruit fly is used in the lab during the study of genetics. That is the study of inheritance and variation. The first one is the female lays many eggs. The female lays many eggs, and as a result of laying many eggs, we will be able to have very many number of fruit fly resulting after the eggs hatches, and therefore if this increases the sample size, and therefore we'll be having a larger sample size of, to study, and therefore this is one of the important why the fruit fly is now or is recently used to study genetics. And the second reason is that have many observable characteristics. And remember, these many observable characteristics are distinct and contrasting. And therefore, this is the second reason why fruit fly is very, very important when it comes to the study of genetics in the lab. When it comes to the study of genetics in the lab. And number 3, the fruit fly is easily bred in the lab. And therefore, it requires minimal requirements. And therefore, this makes it easy for the scientists to reproduce them or to brand them in the laboratory, and therefore at minimal cost. And number 4, it has a short generation time. Actually, the generation, the lifespan of a fruit fly is 10 to 14 days. And therefore, it means that because of a very short generation time, the scientists are capable or are able to study many generations in a given short period of time. And as far as point number 5 is concerned, that is offspring can be backcrossed. Yes, backcrossing refers to a cross between the offspring with their parents, and therefore during backcrossing there will be no harm. And therefore backcrossing can be carried without affecting the F1 the F2 generation, and therefore it means that since the aspect of backcrossing is possible, it makes that the work of the study of inheritance and variation to be successful. And lastly, we have the flies are safe to handle. This is the fact that by the fact that they don't transmit any human disease to human beings, it means that they are very, very safe to handle them in the lab, and therefore this also it is an aspect that enables the fruit fly to be used in the lab when it comes to the study of genetics. Having said that, I'm going to give you a question. I'm going to work out a question here that will help us to understand on the transmission, on the aspect of transmission of genes from parents to their offspring. And the example that I'm going to use in this example here, I'm going to give this example, and the example which I'm going to put, you're supposed to go through this question, and the question reads, there is an example here, an example. You are supposed to using Punnett square, you are supposed to using Punnett square. Determine the genotype, the genotypic should be genotypic, the genotypic and phenotypic ratio of F1 generation between across of pure breed of long-winged fruit fly and another long-winged another long-winged fruit fly in heterozygous condition. What is the solution to this? And as far as the solution to this question is concerned, remember you are supposed to come up with a Punnett square. So I'm going to draw a Punnett square here. The solution to it, I'm going to come up with a Punnett square, and my Punnett square, I'm going to draw it here, and in this example, I'm going to put male, that is the symbol for male, and this is the female. I put those two symbols there so that you can see the symbol for male. Let me put it well so that it can be visible. That is male, and this is as far as the genotype, we are told that it is a pure breed of long-winged fruit fly, and therefore if it is pure, it means that it has two dominant genes, and the two dominant gene, let us use the gene that a dominant gene be L, i.e. that is also to solution, and B, let L be dominant gene, and small l be the recessive gene. And as far as L is concerned, I'm going to put the pure breed, that is 2L. And the other organism, or fruit fly, should have long wing in heterozygous, meaning that there is a dominant gene and there is a recessive gene. And as far as the cross is concerned, if you cross them, you will find that one of the gametes from the one of the parent will fuse with the other gamete from the other parent to form the offspring having this genotype. This is 2 L. And the same case happens to the other one. And in case where the recessive gene is linking or is being crossed with the dominant gene, we'll have a dominant gene, and also there is a recessive gene. So this is capital L, the first one is capital L, and the second one is small l. And the other one, you will have a capital L, and also we have a small l. And therefore, as far as the phenotypic ratio is concerned, phenotypic ratio is concerned, we'll be having the answer is equal to 2. We'll be having 2 long winged fruit fly is to 2 all of them, because sorry, here they are supposed to be 4. All of them have long wings. Why? Because in each and every offspring there is a capital L which represent a dominant gene, and meaning that the characteristic, even if there is a recessive gene, the characteristic of the recessive gene is masked by the dominant gene, and therefore all of them will have long wings, and therefore here we will write 4 long winged fruit fly, and therefore you can see that all of them will be having long wings. And therefore, as far as the genotype is concerned, of F1 generation of that is genotypic ratio. The genotypic ratio is concerned, ratio is concerned of F1 generation, we'll give it as 4, not 4, actually they are supposed to be 2L, 2 capital L, and the other one is 2, 1L and small l. And therefore, as far as the phenotypic, the genotypic is concerned, you will write it as 1 is to 1L is to 1. You simplify, you will get 1 is to 1, L capital and L small. And therefore, as far as this question is concerned, you can see that in our results here, we have two of the offsprings, offspring having having 2 capital L's, and therefore meaning that it is a dominant gene. Both of them are dominant, and therefore, what about the other 2 offsprings? You can see that one of them is having the genotype of one of them, of each one of them. One letter is capital L, that is a dominant gene, and the other letter is a small letter L, which represent a recessive gene. And therefore, when we come to the genotypic ratio, genotypic ratio, not ration, but ratio, you are supposed to write 2 capital L is to 2 capital L's. That is 2. Then you write capital L as per the question, the 1 and 2. And also here we have 2 of them. One, it is heterozygous case. It is heterozygous case such that there is a dominant gene that represent the characteristic of long wings, and also the recessive gene that represent the characteristic of the vestigial wings. However, as far as the second one is concerned, genotypically the genetic makeup, because it has been altered, it means that the genotype will be 2. One of the L is capital and the other one is small, and therefore once you simplify 2 and 2, you are going to get 1 is to 1, 1 is to next one, and that is the end of that question. And as far as the end of that question is concerned, I have come to the end of today's lesson, and I'm going to prepare more lessons, and therefore please keep tuned to my YouTube channel, and therefore please subscribe so that you can always be informed, because biology is life, and therefore it is very, very important as far as living is concerned. You are going to understand the various concepts of biology that will be helping you today as to live in today's life. Till next lesson, bye bye.